This is the third lecture in BIOL1309 Evolutionary Diversity, and it's the first of the recorded lectures. And today I'm going to be talking about the evolution of terrestrial plants. I want to begin by just reminding you of the evolutionary tree or the phylogeny of organisms that I've been referring to in the past couple of lectures. In the past lectures I've been talking about the algae, which you might remember are the three basal branches or clades in this evolutionary tree. Now, the algae were all aquatic organisms. They included the seaweeds, but also included several freshwater aquatic organisms. There were three main lineages of the algae: the rhodophyta, which were the red algae, the phaeophyta, which were the brown algae, and then the chlorophyta, which are the green algae. Now, the chlorophyta are the closest in evolutionary terms to the true plants. The true plants, or the terrestrial plants, also known as the embryophytes, form this very large clade or branch in the evolutionary tree. Every organism that I'm going to be talking about in the remaining lectures in this course are terrestrial plants or true plants. Before I start talking about individual groups of terrestrial plant, I just want to remind you of a few basic features about plants. And I'm going to begin by talking about reproduction. Every plant shows the same basic type of life cycle involving what we call alternation of generations. So this is an alternation between two different multicellular phases within the life cycle. In this diagram, there is this horizontal line. On the left-hand side, we have meiosis, which is cell division, which is accompanied by the halving of the chromosome number. And here on the right-hand side, we have fertilization, where the male and female gametes are fusing together to form a zygote. And of course, the zygote has twice as many chromosomes as the gametes. So within the life cycle, there's a doubling of the chromosome number and then the halving of the chromosome number. And everything above this horizontal line in this diagram is the haploid phase of the life cycle, with only one copy of every chromosome in every cell. And everything below this horizontal line in this diagram is the diploid phase of the life cycle, with two copies of every chromosome in every cell. Now, alternation of generations refers to a life cycle in which there's a multicellular phase of the life cycle in both the haploid and the diploid phases of the life cycle. And the terms we give to these multicellular organisms are the gametophyte and the sporophyte. Now, the word gametophyte literally means gamete producing plant. The word ending phyte means plant, and of course gameto means gamete. So the haploid multicellular individual, shown by these two round balls of cells, are the gamete producing plants, and here are the gametes that are created by the gametophyte. The diploid multicellular phase down here is called the sporophyte because it's a spore producing plant, and here this sporophyte is creating the spores by meiosis. So every single plant has alternation of generations. Every plant has a multicellular haploid individual called the gametophyte and a multicellular diploid individual called the sporophyte. When we look at the phylogenetic or the evolutionary position of true plants, we can see that there's a very close relationship with the chlorophyta, which, if you remember, were the green algae. So the hypothesis is that terrestrial plants or true plants have evolved from ancestors which would have been classified as chlorophyta or green algae. There are lots of lines of evidence which can support this inference. First of all, we're looking at chlorophyll-containing plants. All these plants contain chlorophyll to undergo photosynthesis. They all contain chlorophyll A as the primary photosynthetic pigment, with chlorophyll B and the carotenoids as accessory pigments. You might also remember that when I was talking about the algae, I gave quite a bit of attention to the food products, the starch, which is a primary carbohydrate food reserve, and where it is stored within the cell. What we saw was that the red, brown, and green algae stored the food products in different places within the cell, but that the green algae stored the starch inside the chloroplasts. Well, that is also seen in true plants. And we also saw that the primary component of the cell wall was cellulose in the green algae, and that's something that's shared with the true plants. So there's a lot of evidence to suggest that true plants have a very close evolutionary relationship with green algae. But that evolution of terrestrial plants from green algae involved a very fundamental habitat change. Something very fundamental occurred at this point within the evolutionary tree. And that fundamental change was a change from an aquatic to a terrestrial environment. And if we can imagine the invasion of the land, the major problem that the plants would have been facing would have been one of desiccation, drying out in the air. So before I go any further, I want to spend a little bit of time just looking at what are the adaptations of the terrestrial plants that enable them to avoid drying out on land. And one of the most obvious adaptations is the evolution of a layer known as the cuticle. It's the waxy or fatty substance covering the aerial or above ground parts of the plant. So here's a couple of diagrams. On the right hand side we have a section through a leaf. We have an upper epidermis here in yellow and a lower epidermis down below. But those two epidermal layers are covered by this waxy or fatty substance, cuticle, lying across the top. The cuticle reduces transpiration or reduces the evaporation of water from the aerial parts of the plant. Here on the left-hand side we have a photograph showing a number of plant cells. It's been stained, and this pinkish layer here is the cuticle on this upper epidermis. So one of the main adaptations of terrestrial plants is the evolution of a waxy, fatty substance to reduce water loss. But if you think about the adaptations to avoid drying out in terms of the life cycle, we can see that although it's possible to have a gametophyte and a sporophyte which are both covered in this waxy cuticle, there are two stages in the life cycle which are very vulnerable. The first one is the formation of the gametes and the fusion of the gametes to form the zygote. And the other is the cell which undergoes meiosis to form the spores. Those two stages in the life cycle are very vulnerable to drying out. And if those stages in the life cycle cannot be completed successfully, then the plant obviously cannot survive on land. So let's have a quick look at some of the adaptations to enable those two stages in the life cycle to be completed without drying out. Let's first of all look at the formation of the gametes and the fusion of the gametes to form the zygote. When we look at different groups of true plant, we're going to see the organs which create the gametes. Now, the male gamete, which is called the antherozoid, or the spermatozoid, or simply the sperm, these structures are formed inside an antheridium. So the antheridium is a structure which creates the sperm, and the antheridium serves to protect the sperm from drying out. So if you have a look at this photograph on the right-hand side, you have an antheridium. Basically, this is a bag of sperm here. There is a protective layer covering it, and there's a little stalk at the base. The antheridium is actually a very, very simple structure. It's a protective bag surrounding the cells that form the sperm and then protecting the sperm themselves. Now, the female gamete is the egg, and the egg is protected by an equivalent structure called an archegonium. We'll have a look at the structure of the archegonium in more detail later on in this lecture, but very briefly, here we have an egg sitting inside, in this case, a multi-layered wall of the archegonium. If you look at the top of the archegonium here, there's a long channel or a canal, and this is the canal that the sperm will swim down in order to reach the egg to fertilize it. So the archegonium protects the egg from drying out. But after the egg has been fertilized, it forms the zygote, and then the zygote begins to undergo mitotic cell division to form the embryo. So the archegonium doesn't just protect the egg, but also protects the zygote and then the embryo. So the archegonium actually grows as the young embryo grows and provides continued protection against drying out. If you think about the other half of the life cycle, individual cells within the sporophyte undergo meiosis to produce spores, and they also need to be protected from drying out. Now, most spores have quite a thick spore wall which protects the spore itself. So protection in this other half of the life cycle is slightly less critical. But what's very important is that the cells which undergo meiotic cell division to create the spores are protected. And they're protected by a structure known as the sporangium. And in this photograph, we have a sporangium. It's actually hanging upside down in this particular plant, and later on in this lecture, I'll show you some photographs of that plant. But basically, there is a bag full of spores here. There's a protective wall surrounding that bag of spores, and it's held on a stalk. Now let's have a look at some individual groups of plant. And I'm going to begin by talking about the non-vascular plants. These are plants which do not have a vascular system. In other words, they do not have a xylem and a phloem. So the non-vascular plants form the basal three branches within the true plants. There are three separate groups here: the hepatophyta, the anthocerophyta, and the bryophyta. And I'm going to talk about all three in turn. In the latter part of this lecture, I'll be talking about the hepatophyta, and then in the next lecture, I'll talk about the other two groups. All these non-vascular plants are very, very small. And if the labs are not cancelled because of the virus, I hope you'll get a chance to have a look at some of these plants. Here are some photographs just to illustrate what they look like. These are liverworts, which are very small, plate-like organisms growing over the substrate, growing over the soil or over rocks. They're very, very small. Other plants, like these mosses, are little erect growing plants, often with these curious little spike-like structures, which I'll explain in just a moment. So you will all have seen these type of plants, often growing on tree trunks and over rocks and so on when you go hiking. So what are the characteristics of these non-vascular plants? Well, the most obvious characteristic is that they do not have any vascular system. So vascular plants have a xylem, which is used for the transport of water, and phloem, which is used for the transport of the food products created during photosynthesis in the leaves. These non-vascular plants do not have any vascular system, and that's one of the reasons why these plants are typically very, very small. Now, sometimes these non-vascular plants have cells which have evolved independently of true xylem and phloem, and they perform a similar function. There are hydroids which are involved in the transport of water, and there are leptoids which are involved in the transport of food products. But these hydroids and leptoids have evolved independently from xylem and phloem. They are not true xylem and phloem. They may perform a similar function, but they are structurally quite different. Another characteristic feature of non-vascular plants is that they do not have leaves. Now, leaves by definition have a vascular system. And since we are talking about non-vascular plants here, there cannot be leaves because there is no vascular system. Some of these non-vascular plants have leaf-like organs, but whenever I use that term, I always put it in inverted commas because they are not true leaves. And in much the same way, non-vascular plants do not have roots, because roots by definition, again, also include a vascular system. The root-like organs non-vascular plants have are known as rhizoids. They are involved in water uptake, and they're also involved in anchoring the plant to the substrate. But they are not true roots because they do not have a vascular system. Now, as true plants, all these non-vascular plants have alternation of generations within the life cycle. So just to remind you, this horizontal grey line in this diagram separates the life cycle into the haploid phase and the diploid phase. The haploid phase here, above that grey line, have N chromosomes per cell, where N is the unique number of chromosomes, and the diploid phase has 2N chromosomes in each cell. In other words, 2 copies of every chromosome in every cell. So this type of life cycle is called sporic meiosis. It's shared by every single plant. What is characteristic of the non-vascular plants is, however, that the haploid phase, or the gametophyte, is dominant. And by dominant, I mean it is larger and is longer lived. So if you're out hiking and you see one of these non-vascular plants, the plant you're actually seeing is, in all probability, the gametophytic phase, the haploid multicellular phase of the life cycle. The sporophyte is much smaller, is much more transient, so it's short-lived and is harder to see. So that's a very important point to understand if you're going to realize the life cycle of non-vascular plants. So let me reword that in a very clear way. In the non-vascular plants, it is the gametophytic or the haploid phase that is dominant. The sporophytic or the diploid phase is much smaller. And I'd like to emphasize that the sporophyte actually remains attached to the gametophyte. There's a physical connection between the sporophyte and the gametophyte. And in that sense, the sporophyte can be regarded as being pseudo-parasitic. A parasite is an organism that is dependent on another organism and obtains its nutrition from that other organism. The sporophyte in these non-vascular plants often lacks chlorophyll. It's often unable to create its own food. It obtains its food from the gametophyte, which is why the sporophyte remains attached to the gametophyte. We use the term pseudo-parasitic here because, of course, the sporophyte isn't truly a parasite. It's no more a parasite than a human fetus is that's obtaining nutrition from the mother through the umbilical cord. So let's have a look at the life cycle of non-vascular plants in a little bit more detail. I'd like to emphasize the structures that form the gametes and also the structures that form the spores. So let's have a look at the antheridia, archegonia, and sporangia in a little bit more detail, beginning with the antheridia. So the antheridia, if you remember, are the structures which are creating the antherozoids, or the spermatozoids, or the sperm cells. So they're basically a bag of sperm cells with a protective wall surrounding it. So in this diagram, in this photograph, all the tiny little dots inside are the antherozoids, or the sperm cells, surrounded by the antheridial wall. And down at the bottom there's a short stalk. So antheridia are actually very, very simple structures. We're going to be looking at plant after plant after plant which has a very similar looking antheridium. So you should be able to identify an antheridium really very easily. The eggs, the female gametes, are created in archegonia and are protected by the archegonia. And archegonia are much more complex structures. Here at the center of this archegonium we have an egg, a large cell sitting there waiting to be fertilized by the sperm. And you'll see that the archegonium is actually a flask-shaped structure. There's an enlarged base to the archegonium surrounding the egg. And that basal portion of the archegonium is known as the venter. At the top of the archegonium here we have an elongated neck which has a canal running down it. So that's a long thin tube that the sperm will swim down to reach the egg. The only other structure that's noticeable in an archegonium is a stalk at the base, although in this particular specimen that stalk is very, very short. In others the stalk is rather longer. So the archegonium is a much more complicated structure than the antheridium, and part of the reason for that is that it's not just protecting the egg, it also has to allow the sperm access to the egg, and then after the egg has been fertilized to form the zygote, and the zygote undergoes cell division to form the embryo, the archegonium carries on providing protection. So here's a diagram showing pretty much what I've just said. In the middle here we have an antheridium, and the antheridium is split open at the top here to release the sperm, and these sperm are flagellate, they've got flagellae to swim, and they're being attracted towards the archegonium. So here is a flask-shaped archegonium with a stalk at the bottom. There is the egg sitting at the bottom of the canal, and the sperm will swim down the canal to reach the egg. Now I talk about the sperm as swimming. They swim by thrashing their flagellae. In order to swim you have to have water. Water has to be present in order to swim, and all these non-vascular plants are dependent on water being present for the fertilization to take place. That's another reason why these non-vascular plants tend to be very small. They're small plants very close to the ground, and the water that's present for the fertilization to occur is rainwater. The plants only need a thin layer of rainwater on the ground for the sperm to be able to swim from the antheridium to the archegonium. So, alternation of generations in in the non-vascular plants. Let's have a look at some photographs to show these different phases. In this photograph, there are basically two parts. There's the green part down below, which is the gametophyte. It's the dominant haploid phase of the life cycle. But we also have the sporophyte, the multicellular diploid phase of the life cycle, here in brown. So here are the two different phases of the life cycle, and they're actually attached to each other. It is the gametophyte, which is green, which is containing chlorophyll, undergoing photosynthesis. And here the sporophyte, which is brown, is actually pseudoparasitic on the gametophyte. Let's have a look at the sporophyte in a little bit more detail. This photograph is actually from a different species, but nevertheless, what it shows is a large capsule, which is basically the sporangium. That sporangium is borne on a long stalk or a seta. And then down at the base of that stalk, there's a point of attachment to the gametophyte. So somewhere down there, there's a point of attachment to the gametophyte. Now, I mentioned earlier that there are three different evolutionary lineages of the non-vascular plants, which are collectively often known as the bryophytes. For the remainder of this lecture, I'm going to talk about the hepatophyta, which in English are known as liverworts. There are two main growth forms within the hepatophyta. The most common one are described as being thallose types. So these are flat, plate-like plants growing over the substrate, growing over the soil or over rocks. And this photograph is viewed from above. The other growth form are the leafy types. So they're small, erect growing plants with small leaf-like organs. But please remember, these are not true leaves because they do not have any vascular system. Now in this photograph on the right, the plant is viewed with transmitted light. So light is shining through the microscope through that leaf-like organs. And what you can see is there is no venation pattern. It's not like a true leaf which has a midrib and a secondary vein and so on. There's no vascular system here. There's nothing impeding the transmission of the light. Now I'll talk about both these two different growth forms, but I'm going to spend much more time talking about the thallose type on the left, because this is much more common. So let's have a look at a typical thallose hepatophyta. This diagram shows a transverse section through the plant body. And what you can see is that there are basically three different regions. At the top here, we have this green photosynthetic layer. This is the layer where the cells have chloroplasts, and all these little circles within the cells are the chloroplasts, which of course contain the chlorophyll. Here we have a gaseous exchange pore, enabling gases to enter the plant, and that's where the gaseous exchange takes place across the cell surface. Now this might remind you of the diagrams of a transverse section through a leaf that you will undoubtedly have seen in school biology lessons. And if you remember, leaves have stomata. Stomata are these gaseous exchange pores, normally with a pair of curved guard cells which open and close. This pore is not labeled as stomata, it's just given the generic term pore, because these are not stomata. Stomata invariably have two guard cells which are able to change shape due to the absorption and loss of water, enabling the stomata to open and close. Here, these pores are multicellular. This pore cannot close. So these gaseous exchange pores are always open, and the plant is therefore not able to control or to reduce water loss. So what we find is that these non-vascular plants typically occur in environments which are very damp, very moist, where they're not going to be subjected to too much water loss. So the upper layer of the hepatophyta have these multicellular gaseous exchange pores opening up into these large internal chambers. This is where these chloroplasts are localized. This is where photosynthesis occurs. Down at the bottom of the plant, we have this epidermal layer, shown here in pink, sometimes with these single cell extensions forming these rhizoids, which are the root-like organs anchoring the plant onto the substrate and helping with water absorption. There are also sometimes multicellular scales. The third part of the plant structure, shown here in white, is really just a storage area. Very large, unspecialized cells which are involved in the storage of water and the storage of food products. Now I'm going to give two different examples of thallus hepatophyta. The first one, a very simple one, and then the second one, a little bit more complicated. The simple one belongs to the genus called Rixia. And Rixia is normally a free-floating aquatic hepatophyta. Sometimes, of course, if the water dries out a bit or drains away, the Rixia plant will end up growing on wet mud. And that's what you can see in this photograph. Here we have a single plant of Rixia sitting on top of some wet mud. This plant is actually very, very small. But as you can see, it's clearly a thallus hepatophyta. It has a flat plate-like structure. My next photograph shows a transverse section through one of these Rixia plants. Now this isn't a terribly good photograph, and actually I think it's much easier if we interpret a diagrammatic representation instead. But here in this diagram, we can see the same three zones that I showed you in that diagram a moment ago. Across the top here, we have the chloroplast containing cells. So each of these cells here has small chloroplasts inside. We have gaseous exchange pores. But because the Rixia plant is very, very small, these gaseous exchange pores are compressed together. So basically we've got a whole series of very, very closely positioned gaseous exchange pores. Down on the bottom of this plant we have the lower epidermis. Again we have the rhizoids and sometimes multicellular scales. And this region here is the region of large unspecialized cells involved in water storage and food storage. Now the plant we're looking at here, of course, is the gametophyte. It's the multicellular haploid phase of the life cycle. And gametophytes are the gamete-producing plants. So in this diagram, we would expect to see antheridia and archegonia. And if you look very closely, you can see that. Here we have an antheridium. It's a bag full of sperm cells. It's embedded in the upper surface of the gametophyte. And over here, we have an archegonium, a flask-shaped structure with the egg at the base, at the bottom of a long canal. My next slide shows these structures in a bit more detail, the antheridium on the left and the archegonium on the right. Now this diagram of an antheridium actually shows a rather immature antheridium. These pale grey cells at the center are not yet the sperm. They're labelled fertile cells. They're the cells which will undergo cell division to create the sperm. But the archegonium on the right-hand side is much more well developed. There's an egg sitting, waiting to be fertilized. And as we've seen, the sperm or the antherozoids, when they're released from the antheridium, will swim through rainwater towards the opening of the archegonium, swim down the neck or down the canal to reach the egg. Now the second example of a thallus hepatophyta that I want to talk about is a much more complicated plant. It's a plant called Marchantia, and if the labs go ahead, you'll be able to see Marchantia in the flesh. We can tell it's a much more complicated plant just by looking at this photograph. Although down here on the left-hand side of the photograph, we can see the flat plate-like plant body characteristic of a thallus hepatophyta. We can also see these rather complicated structures here looking like little umbrellas raised up above the plant. These umbrella-shaped structures are specialized structures bearing the antheridia and the archegonia. So let's have a look at those in a little bit more detail, beginning with this photograph here. Here we've got a whole series of structures which are actually called the antheridiophores. Now the word ending phore, P-H-O-R-E, means a structure which is bearing something else. So this antheridiophore is clearly a structure which is bearing the antheridia. So what we're looking at in this photograph is a male gametophyte, a gametophyte which is only producing antheridia with sperm inside. Here's a diagram showing the same structure, this umbrella-like antheridiophore growing from the upper surface of the thallus gametophyte. Now my next slide shows a longitudinal section through one of these antheridiophores. And in fact, the structure of the antheridiophore inside is no different from the structure of the normal thallus hepatophyta. Let me first of all bring your attention to the main plant body down here. Here we've got the chloroplast containing upper layer with their multicellular gaseous exchange pores opening up into these large internal chambers, with the lower epidermis down below with the rhizoids and the large storage cells in between. This structure here, of course, is the antheridiophore itself, and in fact it's identical to the normal plant body. Here we have the antheridia embedded in the upper surface of the antheridiophore, but the antheridiophore has all the other features I've just talked about. We've got multicellular gaseous exchange pores opening up into these internal air chambers. We have a lower epidermis down here with these rhizoids and scales, and we have the storage cells in the middle. So basically, an antheridiophore is just a localized area of the plant body raised up on a stalk or a seta. Now the purpose of raising the antheridia up high is to aid dispersal of the sperm. By holding them up above the ground, the sperm can be dispersed more distantly. But please remember, we're looking at very, very small plants here. Every year, students turn up at the labs and are very disappointed to see Marchantia because the height of this antheridiophore is maybe about 1 centimeter. So don't imagine that these are large palm-like trees being blown around by the wind. These are very, very small plants. Everything is on a very, very small scale. Now let's have a look at a female gametophyte. So here we're looking at a gametophyte of Marchantia, and it's bearing slightly different structures, again looking a little bit like umbrellas, but this time umbrellas where the fabric has been lost between the metal struts of the umbrella. And these structures are bearing the archegonia. So they're called archegoniophores, structures bearing the archegonia. Again, here we have a diagram showing that. So there we have an archegoniophore born on a long stalk from the upper surface of the thallus hepatophyte. And here's a longitudinal section. We can see that we have a series of downward projecting fingers or arms. And here we have structures which I hope you're now able to recognize as archegonia. There is one of those archegonia blown up rather larger to see. So these archegonia are actually suspended upside down from, if you like, the armpit of these downward projecting arms. So the egg would be located here, inside the venter, and here's the long neck with its canal actually hanging upside down in this plant. Now we have a series of photographs here showing what happens to the archegonium after fertilization. Here we have an archegonium which has contained an egg which has been fertilized. It's already formed the zygote, and the zygote has now started to undergo mitotic cell division. So we have here a ball of cells. It's a very young embryo, part of the diploid sporophyte generation. This layer of cells surrounding it is the venter of the archegonium. So the venter is actually growing to protect the young developing ball of diploid cells. And if we have a look at a later stage, here the sporophyte has actually become much larger. We can see we have cell differentiation, different types of cell located there. The venter has got much bigger again, and the neck with its canal is beginning to shrivel up and dry. And if we have a look at a much more mature individual, here we have the mature sporophyte. So we have a sporangium, which is just a bag of cells born on a stalk or a seta. And then that dark structure at the top there is the foot, which is remaining attached to the gametophyte. Because all these cells at the very top of this photograph, don't forget, are part of the gametophyte, the sporophytic generation. So the archegoniophore has archegonia which are elevated up above the ground. But this is not because of dispersal of the egg, because the egg is not dispersed at all. The egg remains in the archegonium, waiting for the sperm to arrive. The reason why the archegoniophore is elevated is because this sporangium, with its spores, will develop where the archegonium was. So the archegoniophore is holding the sporangia up high, and of course that enables better dispersal of the spores from the sporangium. Now if you have a look inside the sporangium, we can see something very interesting. This color photograph shows the contents of the sporangium. We have a whole series of very, very small blobs here. Some are green, some are red. These are all spores waiting to be dispersed. Whether they're green or red is just whether they've picked up the stain or not. But what's very interesting in this photograph is not the spores themselves, but rather these elongated structures which are embedded in amongst all the spores. These elongated structures are called elaters, and if you look very carefully you can see that each elater has a rather spiral wall thickening. And it has spiral wall thickening because it will change shape as it dries out. When the sporangium reaches maturity, it will begin to dry out. As these elaters dry out, they will suddenly change shape, and this will have the impact of pushing the spores apart and even throwing them out from the sporangium. So here's a photograph of an archegoniophore. In this particular case, the arms of the archegoniophore are actually very short, which is very good because it shows the sporophyte much more clearly. So there's a sporophyte. This brown structure is a sporangium, and the white structure there is the seta or the stalk, keeping it connected to the gametophyte. But here on the left, we have a sporangium, which is split open. So all these pale brown structures there are the spores, and they're held together by these elaters. And these elaters will be changing shape as they dry. And a very sudden, very rapid change of shape will have the impact of throwing the spores out from the sporangium. So what I always like to do is to talk my way through a life cycle for the individual plant I'm talking about. So here is the life cycle of Marchantia. So here we have a horizontal line. We have meiosis on the right-hand side. We have fertilization on the left. So everything below that horizontal line is the haploid phase of the life cycle with N chromosomes. In other words, one copy of every chromosome in every cell. And everything above the line is the diploid phase with 2 N chromosomes, or 2 copies of every chromosome in every cell. So let's talk our way through the life cycle, beginning with the gametophytes. So the gametophytes are these flat plate-like structures. We have separate male and female individuals. The male individual has these male antheridiophores, and each antheridiophore has the antheridia embedded in the upper surface. The female individual has similar archegoniophores, which have the archegonia in the armpits of these downward projecting arms. The antheridia will release the sperm, which are flagellate. They'll swim through rainwater. They will swim down the canal of the archegonia and fertilize the egg. So we then have the formation of young sporophytes. And the sporophyte has a 3-part structure: sporangium on a stalk with the foot remaining in contact with the gametophyte. And the spores develop inside the sporangium. The spores develop as a result of meiosis, so these spores can be drawn below that horizontal line. These individual spores are wind dispersed, and after they settled, they will begin to germinate and will produce the new gametophyte. Now, before I begin, I stop talking about Marchantia. I just want to say give you a bit of information about asexual reproduction. Because several of the photographs and the diagrams I've shown you have included these structures, which are called gemma cups, small cup-like structures on the upper surface of the thallus plant body. And if you have a look inside these gemma cups, we can see small structures which are known as the gemmi. I think they're clearer in this photograph. Here we're looking down on top of the thallus plant body. This entire round structure is a gemma cup, and inside we have gemmi. And the gemmi are packets of vegetative plant cells. And the gemma cup functions because as a raindrop falls into the gemma cup, the gemmi will be splashed out. So the act of raindrops falling into the gemma cup will serve to disperse the gemmi away from the parent plant. And each of those gemmi can germinate to produce a new gametophyte. And I'd also just like to point out all these small circles here on the upper surface of the gametophyte. These are the openings into these internal air chambers. Now, I mentioned earlier that there were 2 different growth forms of Hepatophyta, and I've spoken at length about the thallus types. Very, very briefly, I want to just show you some photographs and diagrams of the leafy types. So the leafy types have small erect growing plants with leaf-like organs. This is a genus called Frulania, although the name doesn't matter too much. if you have a look in the textbook, you'll see that there are some specialized structures shown by these leafy hepatophyta. This diagram shows two structures labeled here, the perianth and the andrecium. Now, both these terms are actually applied to different parts of a flower. So when we're talking about flowering plants later on, we'll encounter these terms again. But the perianth and the andrecium, in the context of the leafy hepatophyta, are leaf-like structures which are wrapping themselves around the antheridia and the archegonia. So the perianth here, if you were to peel back those leaf-like structures that form the perianth, you would find the archegonia inside. And if you peel back the leaf-like structures of the andrecium, you would find the archegonia inside. So within the context of the leafy hepatophyta, the perianth and the andrecium are serving to protect the archegonia and the antheridia from drying out. And that's the end of this lecture. In the next lecture, I'll talk about the two other groups of non-vascular plants.